talk about human disease caused by bacteria. We're going to finish these slides, and then I'll go back to talk about a little bit after class reading, and maybe we talk about 2 case studies. Okay? So first of all, we talk about upper respiratory tract and the lower respiratory tract disease, major pathogens, bacterial pathogens. Today we're going to talk about human digestive tract. Okay. Here are so many things we want to mention. These one by one. First of all, oral cavity. And we mentioned there's a lot of bacteria in the oral cavity. We call them a Viridium streptococcus, like Streptococcus bovis, Streptococcus mutans, and the Streptococcus vitis, a lot. Okay, then we have esophagus, then go to liver, go to stomach. Now you should know, stomach area should be very few bacteria. Why? Because low pH. Extremely low pH. And that pH is around 1 to 1.5. That extremely low pH will not let most of the bacteria and the microorganisms survive. There is only exception. Helicobacter bacteria, we call it HP. We will mention this bacteria is survived that possibly could cause stomach cancer because they can hydrolyze ureas, become ammonia. We will mention this in the lab. Okay? Urea become ammonia, and this ammonia could buffer the low pH back to 4 to 5. So Helicobacter pylori can grow there, can survive. Now you move on to intestinal and small large intestinal. There will be a majority of the bacteria reservoir. Okay, so when we go to small or large intestinal, this will be a huge microbial reservoir. Could be up to more than 10 to the 10 cells per gram if you get from the fecal sample. This is a new theory. People think about our intestinal gut microorganism is a major contribution for our body, as important as our brain and our heart. That's a new theory about that. You know, lactic acid bacteria and some other beneficial bacteria, bifidobacteria, but this reservoir play a key important role for our physiology. Therefore, there's a lot of technology talk about it. For example, you heard about 16S RNA, biotechnology. Tons of research are doing that. They think this is a new center of our body. Okay, just something I want to let you know. Besides, some of the bacteria in there is anaerobic. A little bit aerobic. Some of them in the middle is facultative. So this is just give you an overview of microorganisms in the human digestive tract. These slides I just mentioned which is almost what I wrote here, and to give you the conclusion. So look at the stomach. Very few, 100 cells per ml. So make sure in this class, in the lab, we talk about is the concentration, not the total amount. Okay? And you see small intestinal, 10 to the 4 cells per ml. Large intestinal, huge. Could be up to 10 to the 12 cells per gram. And the majority of them, anaerobic bacteria. The ratio between anaerobic to aerobic is 10,000 to 1. And if you get a fecal sample, there is lots of research going on right now, like we call it a translation medical research. So for example, you are feeding babies, newborn babies, dairy products or lactic acid products. Then you periodically to get the fecal sample from the baby. And then you do analysis to see the bacterial community in those type of the fecal sample. Then do some of the cluster analysis to see the similarity. All those things tons of research are doing right now. Because our fecal sample, about more than 30% is bacterial cells. Okay? And traditionally, we think that's our immune system. Same as important as your white blood cell. So just give you an idea. The normal flora of the alimentary tract. This is slides, tells you almost everything, a conclusion. So we're going to talk about today, major pathogen happens there. The first slides of what we talk about here. We will be introduced to two different concepts, which is food intoxication versus food infection. Okay, first of all, give you some of the example. What is the example for the food intoxication? We talked about Staphylococcus aureus, Clostridium, and maybe Bacillus. But some people say Bacillus subtilis is normal, is untoxic. There's some research going on about it. Just an SPP. Okay, what is a food infection? You heard about the news already, Salmonella outbreak. E. coli 0157H7 and Listeria monocytogenes. These are, we call it a food infection. Okay, what's the difference between that? This is the key. Food intoxication, you are eating toxin. Food infection, you are eating bacteria. What type of the toxin are these? Major or majority is enterotoxin. Now when you're eating bacteria, of course bacteria will generate toxin. Majority of them are endotoxin, especially those gram-negative bacteria, SRA, lipopolysaccharides. Now, what's the major difference between that? Enterotoxin is heat resistant, and endotoxin is heat sensitive. So, what this means for preparing the food? For food intoxication, you'd better have to do a good job of pre-harvest or food handling. And cooking may not work sometimes, especially Clostridium. You have to do a sulfur cooking. Just the heat it may not work. For food infection, Let's say salmonella in a poultry meat, E. coli in ground beef patties. You do a cooking, which means you do a heating, can solve the problem as long as you're curing the bacteria. Okay, so that's a major difference. Another thing I want to mention, but it's not really true sometimes, is the onset time. The onset time for food intoxication is very short. Can be as short as like 4 to 6 hours. And this guy usually 6 to 12 hours. So a little bit long, but not really. So lots of the book will tell you the onset time for food intoxication is very quick. Let's say I mentioned a chicken salad contaminated with Staphylococcus aureus. You could have vomiting, diarrhea, abdominal pain, nausea about 5 to 6 hours after you eat a Staphylococcus aureus contaminated chicken. Okay? So these are the major things difference from here. Here you can see the slide. Staphylococcus aureus, onset time 1 to 6 hours. Nausea, vomiting, diarrhea, Clostridium botulism. We're going to talk real quick. Double vision, difficult swallowing, breath problems. And the Bacillus cereus, vomiting, abdominal pain, diarrhea, nausea. These happens real quick. Could be as long as short, maybe 1 to 6 hours. So this slide gives you example is food intoxication. And the food infection, we have a slides later on will be show you is actually right later. This is a food infection. Okay, we're not going to mention that too much because that's belonging to another class, which is food microbiology. So that's why I want to just very briefly go over the difference between food intoxication and the food infection. Okay, now we got to spending majority of the time today. We will talk one bacteria group or one major bacteria in the food poisoning, or we say food intoxication, which is Clostridium. Okay, so we will go over very detailed for everything, pretty much cover majority of today's class. So we want to talk about that. First of all, Clostridium. We mentioned this is the endospore-forming bacteria. So number one, we want to talk the whole picture. What is Clostridium? This is gram-positive. Large shape, a little bit curved. Not too much, a little bit slightly curved. Endospore-forming bacteria. And very important, streak anaerobic bacteria. Okay, remember, bacteria could be differentiated into aerobic, facultative, which means they grow in the presence, absence of oxygen, but grow better with oxygen. That is the majority of our bacteria. And the anaerobic bacteria, streak anaerobic bacteria. Why? Remember the question in the examination one? No. SOD. What is that? Sulfide dismutase. No catalyst. Which means if the oxygen is presented, the bacteria do not have the enzyme to hydrolyze those toxication materials. Okay? So that's the first thing we want to measure. Second, what are the Clostridium? We will talk. We will talk Clostridium, lining, Clostridium tetanus, Clostridium perfringens, and the Clostridium difficile. We will talk all these one by one in very detail. Okay, first of all, Clostridium botulinum. Clostridium botulinum, we also call it botulism. Will cause a problem is botulism. Okay, what is botulism? There is a toxin comes out. Okay, there's a toxin comes out, which is also an AB toxin. Sorry, this is I messed it up here, sorry. A, B, C, D, E, F toxin in the United States. ABE is responsible for 60 to 80% in US. Okay? What type of the toxin really going to cause a problem? What type of the problem will have? Neuroparalytic. What are they? Difficult speak, difficult swallowing, double vision, and I will say quotation mark, surrounded muscle weakness. So neuroparalytic symptoms. This is the overall. Now we want to talk about the different type of the botulism. What are they? We want to mention these one by one. Okay, B. First of all, we will talk about 3 different types of the botulism, which is wound botulism, infant botulism, and the food botulism. So we talk about these one by one. This slide here is wound botulism on the side. Okay. Wound botulism is very simple. On your skin surface, you have cuts, you may have bites, you may have surgery, or surgical wounds, or some people has needle injection. What are those people? Frequently to do it, drug abuser. These are the places could cause wound botulism. The reason is on the skin surface. Let's say this is skin. You have the small cuts. This area, very, very low oxygen. Clostridium is an anaerobic bacteria. They could survive there and create a toxin and spread it into your body, cause you muscle weakness. So that is wound botulism. Very simple. Okay, in the real life, the major risk of people is actually drug abuser. So we're not going to mention this too much. Second one is infant botulism. Okay, for infant botulism, you're going to have to remember one sentence will let you understand. Is never feeding newborn baby, or we say any baby less than 1 year old honey. Otherwise, you have floppy baby. Look at the picture in the slides. Why? Muscle weakness. Okay, then the question, why is it will cause muscle weakness? Okay, remember, the muscle, you will have relaxing and contraction. Okay, this is a little bit of anatomy, is that right? Contraction and relaxing. What are the neurotransmitters control them? Which one is this one? What is that one? Glycine. What is that one? Chlorine, is that right? Or we say acetylchloride. Okay, for newborn baby, if you eat honey, the honey more or less has some Clostridium. They originally coming from where? Soil or something. For newborn babies, the normal bacteria has not been established. If they go to intestinal, in that environment, remember, intestinal area, anaerobic bacteria is dominant. They will start to have a sporation. The dominant there, transfer toxin. This botulism toxin will be blocked, azidyl chloride. So the muscle will not be contracted. Therefore, it's totally weak at the relaxing muscle. Therefore, it's floppy baby. Okay? So that's very easy to understand. Now, we want to spend some time talk about the last one. Our left today. Last one we want to talk about is food botulism. Okay, look at the picture on the top. What are they? This is extremely popular in the West Virginia rural area. Canned food. Okay, what are the canned foods? Let's say you're gonna tint that part. Okay. What are they gonna put there? Could it be corn, tomato, bean, or beef stew? Some people put everything there. Where do they come from? Soil. And I want to mention Clostridium is originally major coming from soil. They are not really in purposely to attacking us. But if you don't do a good job, they will going to hurt you. Okay, the soil. So you add it there. Okay, you let them grow. These type of the canned food is favorable for Clostridium to grow because they call it low acid foods. What are the low acid foods? pH around 4.6. Water activity around 0.85. Some of the textbooks say this is larger than 4.6. That's called the low acid foods. These type of environment, most of the other bacteria, or we say vegetative cells, let's say generic E. coli, they will be killed. Eastern fungi, some of them will be killed, some of them might be survived. But Clostridium will be dominant. Okay, so there is a common mistake people making. In the old times, they do cold pack for these canned food. How they do cold pack? They boil in 2 hours. Okay, then immersing in cold water. Then they can. That's not good. Boiling 2 hours, not going to kill endospore, no infection for endospore. And only going to kill vegetative cells. Even if the endospore bacteria are dominant. So this method is bad, really bad to do. Now how we do it? In some rural area, in the traditional methods they do, you have to add lemon juice, some of you. Some of them add vinegar. They try to lower down the pH before or a lower 4.0. And I tell you one thing, this is the standards used by Department of Agriculture, West Virginia. The regulation for canned food is the pH lower than 4.0. We have to do some of the research or extension activity to verify that. But this is the current standards they're using. And they think Clostridium could be almost or inhibited. Now, for some of the large scale industry, what they do? Pressure cooker. The pressure cooker, look at the picture there. What's the condition? PSI 15 Pascal. At 121 degrees Celsius, at least 15 minutes. This is going to be killing endospore. Okay, so this is the methods, what they are using. Now, for canned foods, we mentioned about, in the United States, ABE is a major toxin. This one, typically for canned fish, E toxin. So, somebody, if you get contaminated, you really have Clostridium botulism, what you have to do? You need using antitoxin immediately.